Hello, everyone. Welcome back to my single-cell RNA sequencing data analysis. So Seurat is one of the best packages that we use it for single-cell RNA sequencing data analysis. Recently, Seurat updated to version 5. Because of the package update, lots of people encountered errors or have different issues. So I'm going to make a new videos and show you how to use the Seurat v5 version. So first we can load the packages Seurat, Tidyverse, Patchwork, and we also set a set for code reproducibility. So normally we run the session information after we finished our analysis. So today I'm going to run session information first in order to show you the versions of my Seurat. You can see in my computer I'm using both Seurat and the Seurat object as 5.0.1 version. In nowadays, single-cell RNA sequencing data analysis is widely used. It is very rare for you just analyze one single-cell RNA sequencing dataset. But some of you asked me to demonstrate the analysis in V5 for just one dataset. So today, I'm going to show you how to analyze one single-cell RNA sequencing data in Seurat V5. This video will be very useful for beginners to understand the Seurat standard workflow. So again, I'm going to use one single-cell RNA sequencing data from the GSCE 132771 dataset. Because this dataset was generated by 10 times genomics technology, so we can use read 10 times to load the data. Let's run. So you can see we loaded the data as large DGC matrix. Then we can use the large DGC matrix data as count to create a Seurat object. The project name will be GSCE 132771. We keep genes that express at least in 3 cells, and we keep cells with at least 200 gene expressions. So now we can create the Seurat object. You can see we have a Seurat object here. Now we can use the percentage features function to calculate the percentage for mitochondria genes. We can do the calculation. So after that, we can run a wiring plot for the M features RNA, N count RNA, and percentage mitochondria DNA to decide the parameters for subset the data. Let's run the wiring plot. You can see we generated 3 wiring plots here. You can see for the M features, we can keep cells with M features RNA below 4,000 and the N count RNA below 20,000. And we are going to keep the mitochondria DNA percentage below 10. So now we can subset the data. After that, we can have a look at the metadata. Let's run the view function. You can see the row names are the barcodes. We have the origin ident as the GSCE 132771 because we set it as the project. Then we have the N count RNA, M feature RNA, and the percentage mitochondria DNA. So we performed the quality control step. Let's have a look at the class of this Seurat object for the RNA. If we run, you can see it is a C file. That means we are doing the analysis in the Seurat v5 version. Now we can have a look at the Seurat object. You can see in this object we have 3,616 cells, and we have 17,574 genes. At the moment, the variable features is 0 because we haven't do the analysis yet. So we can click the Seurat object and have a look at the data structure. Let's click it. You can see here is the Seurat object name. We have the assays here. You can see it is assay RNA. In the RNA, we have the layers. You can see at the moment we have the counts. So now we can do the analysis. So first we can normalize the data. Then after that we find the variable features. So now we can scale the data and run PCA. So now we can run the elbow plot function to determine the best pieces for downstream analysis. We can run the elbow plot now. You can see I set the NDMs as 50. We generated the elbow plot. You can see the elbow shape is somewhere here. So from the elbow shape, we can consider this area is the forearms. Any PC numbers on the forearms will be okay for you to use for downstream analysis. If you choose a PC before, for example, here 10, then you cannot capture all the information from this dataset for downstream analysis. So here I'm going to choose 20 for the demonstration. We can use the first 20 pieces to find the neighbors. So after that, we can find the clusters. I set the resolution as 0.2. You can see with the resolution at 0.2, we have 14 cell clusters. Now we can run UMAP. So we finish the Seurat standard workflow. Then we can have a look at the metadata again. You can see now, after analysis, we add the cell cluster numbers for the RNA resolution 0.2 and the cell cluster number are the same as Seurat clusters. And also we can have a look at the data structure again. You can see now in the 6th data slot, we have the RNA. If we click the RNA, we have the layers. Now we have 2 layers. You can see before we only have the counts. Now we have the data layer. This is the normalized data. And also we have the scaled data here. If we scroll further down, you can see the reductions. We run the PCA and the UMAP analysis for this data set. So the data structure looks the same as Seurat v4 because here we only have one sequencing dataset. Seurat v5 was designed to do better data integration analysis. So if you only analyze one single cell RNA sequencing data, there's no difference on the data structure between v5 and the v4 version. So now we can run the DIM plot to see the cell clusters. You can see we have 14 cell clusters here. We can run the feature plot because this is a data set from human lung. We can use 4 classic mark genes to identify the non-cell types. Let's run the feature plot first. we can zoom in. You can see the collagen 1A2 identify mesenchymal cells, EPCAM labels epithelial cells, and the CLDN5 labels endothelial cells, PTPRC labels immune cells. So after the feature plot, we can find the markers for different cell clusters. For example, I'm going to demonstrate how to use find the markers function to identify markers for the cell cluster 1. Here we just need to set the identity 1 as 1. Let's run the find the markers function. You can see we generated the mark genes for cluster 1. We can click the data frame and have a look at the mark genes for cluster 1. Because the cells in cluster 1 are epithelial cells, we can see mark genes, for example, MUC1, KRT19. Down here we have the ESCFTPA2, ESCGB3A2. They are mark genes for non-epithelial cells. Okay, that's today's analysis. I showed you how to analyze just one single cell sequencing data in Seurat V5. We went through the standard workflow analysis. You can see there's no difference for the analysis between Seurat V5 and the V4 version if you just analyze one single cell sequencing dataset. In my next video, I'm going to show you how to analyze more than one single cell RNA sequencing dataset. So I hope to see you in my next video tutorial.